Hi, it's Miss Parrott, and this is the second video in my series AP Biology Equations Explained. Today we're going to be looking at mean, median, mode, and range. In AP Biology, it asks us to be able to use those four statistics to describe data and to interpret experimental results. So the goal here is to help us use these measures to summarize and understand the main features of a data set, whether we see that in a table, a graph, or a described experiment. First, let's look at the mean. Now, the mean is on the AP Bio formula sheet in two different ways. At the bottom, they just say the word mean, and they say it's the sum of all data points divided by the number of data points. And we're pretty comfortable with that. But at the top of the sheet, the very first equation you see for mean, which is very simple, looks like this. And that's scary, and it has a lot more variables than you thought it should have, and symbols you may or may not have ever seen. So quickly, I want to demystify that, and then let's just do a quick practice. So mean is represented by the variable x bar, so x with a line over the top. N is the number of data points, or the number of individuals. And then this big Greek letter is sigma, that's a capital sigma, and it means sum, or you add them all up. What are we adding all of them up? We're adding up all of the x sub I, all the individual x's, or all the individual data points. So this does mean, this whole funky part is that whole add up your data points, and the 1 over n is and then divide by how many data points you have. And that will give you your mean. So I just don't want you to be scared there. Here's an example of plant height. So let's say that we had two groups, an experimental group and a control group. The experimental group, we gave fertilizer and waited 2 weeks for it to grow. And these were their heights. 12 centimeters, 15, 13, 16, and 14 centimeters. So our first step, right, is to add all of those up and then divide by our n. N is the number of data points, and we have 1, 2, 3, 4, 5. Okay, so there's 5 there. When we sum or add up the top row, we're going to get 70. 70 divided by 5 is 14. 14 centimeters is our mean. Our average, the central point. Next is median. Median is often ignored, but it's very important, especially in biology. But this is your middle ground. The formula sheet quotes it as saying it's the middle value that separates the greater and lesser halves of the data set. So it's the middle number. This is especially useful when you have extreme values or outliers. And if you are any individual doing any kind of lab work, there's almost always going to be some number that's just off. So using median instead of mean is a good opportunity because it keeps that number that's very extreme from skewing or impacting the whole data set. So let's take our same plant height data set and add one that grew 30 centimeters in 2 weeks. That is almost twice the mean. That would throw off the mean and make it much higher and not really representative of the data set. So how do I find the median? First, you're going to order your data from least to greatest, right? It tells us our lesser and greater halves. Now, to find the middle number, so if your data set has an odd number of values, or n is odd, then you're just going to cross out, like line them all up, cross out till you get to the middle, and whatever the middle number is, that's your median. But if in this case you have an even number, you're going to do just like before, cross out your least and greatest, or greatest and least, and now I'm down to 2 middle numbers, 14 and 15. Add those two up and divide by 2. So 14 plus 15 is 29 divided by 2. Our median is 14.5 centimeters. So even though 14.5 isn't in the data set, it's still the median. Next, we have mode, the most frequent. This is the value, this is the quote from the formula sheet, the value that occurs most frequently or most often in a data set. One thing I encourage you to do is go through that whole formula sheet, and if there are words like frequent that you're not familiar with, just like look it up now before the exam so that you are prepared for that. So let's say we did a different plant growth experiment, and in this one we were counting growth by the number of leaves that each plant produced, and then we tallied up the data. In this case, 3 plants had 8 leaves. And all the others had 2 or 1. So 3 plants had 8. 8 is the mode of the data set. Now we have to remember, mode's a little different than the others because you can have multiple modes. So let's say there was another one with 3. Let's say, then you would have a mode of 8 and 10 because they both had 3. They both had the highest. Now you can, so that's multiple modes. You can also have no mode. So if every plant had a different number of leaves, this data set would have no mode. We use mode when frequency, so things like allele frequency, matter. All those other ones looked at central tendency, or they are used to describe what's the most common, or they're all ways of taking the average. Range, on the other hand, is a measure of variability. The range is the value obtained by subtracting the smallest observation, or sample minimum, from the greatest, or sample maximum. So you just take your highest value and subtract from it your lowest value. If this is our same data set we had before, our max value is 16 centimeters, our min value is 12 centimeters. 16 minus 12 is 4 centimeters. That is our range. Range is simple, but it's important to help us realize how precise our information is, and if our data is highly variable, we can try and think about why. Or if the range is very small, then we have a high level of precision, and perhaps we can have greater confidence in that data. Now, Miss Parrott, last time I checked, I learned how to do mean, median, mode, and range in middle school. And that may be true. But in AP Biology, you not only have to know how to calculate it, you got to know when to use what and in what biological context that makes the most sense. And then be able to, in writing, justify why you chose that measure and what information it gives you about the experimental results. That's where we're taking it to the next level. So when we're interpreting data, something like a large range could suggest high variability and low precision. This could mean some inaccuracy in your experimental planning or execution, or it could just mean inherent to the variable you're looking at, that high variability is part of its essence. If the mean of your experimental group is significantly different than the control group, then that variation in the mean could suggest that the treatment you did in the experimental group had a major effect. And if the mean is very different from the median, if those two values are very different, that might suggest that there were some outliers or some extreme values that need further investigation. Now, how is this showing up in the AP Bio exam? These types of questions can be in the multiple choice section or in the free response section. So you need to practice writing out descriptions, comparisons, justifications for experimental results. So one of the things you're going to be assessed on is Science Practice 4, representing and describing data. So what? You have a data table. What does this mean, right? You need to practice writing that before the exam. Science Practice 5, statistical tests and data analysis. So you know how I talked earlier about if they are significantly different, what does that mean? So you're going to have to be able to use things like mean. So you got to calculate mean in the first place to determine other values like standard error of the mean. You're going to see these things in real world context. Say like calculate any of those 4 measures for plant growth, or compare the mean and median of the times it took this enzyme to convert all the reactants to products, or identify the mode number of offspring. These are the type of context you're going to see on the biology exam, AP Bio exam. You're absolutely going to see this setup in data tables and in graphs, and you're going to have to interpret those as well. And then you're going to have to justify which statistic you're using to describe your data and why you chose that. I chose mean over median because my data was very consistent and I didn't really have any outliers, things like that. So that about wraps it up for mean, median, mode, and how you're going to see those in range and how you're going to see those on the AP Biology exam. Coming up next, using something like mean to help us do more complicated interpretive statistical tests like standard deviation and standard error of the mean to determine statistical significance. Remember to like and subscribe to this channel, like the video. There will be links to practice for both calculation and interpretation of data in the description box below. Look forward to seeing you next week.